Welcome to Proteins from DNA Sequences by Maddie Maxwell. A protein that is 5 amino acids long is formed from this DNA sequence in vivo. We must find which DNA strand contains a protein that is 5 amino acids long. For the 5' to 3' strand, or the orange strand, we need to look for start and stop codons. Here are the start and stop codons. At the beginning of the orange strand, ATG is the start codon, which codes for MET. The stop codon is located further down at TGA. This protein is only 4 amino acids long. For the 3' to 5' DNA strand, or the green strand, we must flip the green DNA to be able to read it. This is the green strand flipped to 5' to 3'. For the start codon, ATG is marked, which codes for MET. Further down, TAA is marked as the stop codon. This green DNA strand has a protein that is 5 amino acids long. Which is the template strand and which is the coding strand? The orange strand's protein is only 4 amino acids long versus the green strand's protein being 5 amino acids long. The orange strand is our template strand and the green strand is our coding strand. We will be using the coding strand or the green strand to demonstrate point mutations in this video. For point mutations, here's how to convert DNA strands to mRNA strands. Using the green strand, you must convert thymine or T to uracil or U. Once the green strand is converted to mRNA, you can code for the DNA strand. This codes for MET, VAL, ARG, FET, LIS, and the stop codon. This is called the amino acid sequence. For the first point mutation, we are discussing missense point mutation, which is the change of a single base pair which causes a different code for the amino acid sequence, resulting in a different protein. I changed AGG to ACG for mutation, which we need to convert this DNA sequence to mRNA. Here's the mRNA strand for the missense point mutation. Once we code this mRNA sequence, we can see that ARG is changed to THR due to the mutation of the guanine or G to cytosine or C. This is a transversion since guanine or purine is changed to cytosine or pyrimidine in the green DNA strand. The second point mutation is a nonsense point mutation, which is a change in the DNA strand that causes the end of the translation earlier than the original translation. Here I changed TAA to TAG, which is a stop codon. After converting to mRNA, we can code the green strand and see that the change from adenine to guanine codes for a stop codon and ends the translation early. Here's the new amino acid sequence due to nonsense point mutation. This is a transition as adenine, a purine, is changed to guanine, also a purine. The third point mutation is silent point mutation, which is a change in a single base pair but does not cause a different code for the amino acid sequence, which results in the same protein as is expected. Hence why it is called silent mutation, since it is not noticeable to the amino acid sequence. Here I changed AGG to AGA, which codes for the same. After converting to mRNA, we can code the mRNA and see that the change from guanine to adenine codes for ARG, which is the same as before. Here is the new amino acid sequence. This is a transition as guanine, a purine, is changed to adenine, a purine. The last point mutation is a read-through mutation, which is the change of a stop codon single base pair, which causes a non-stop codon and furthers the amino acid sequence longer than expected. Here I changed this single stop base pair, TAA to TCA. After converting to mRNA, we can code the green strand. The amino acid sequence is now longer than expected as the stop codon was changed to a codon that does not code for stop. The amino acid sequence is MET, VAL, ARG, FET, LIS, and SER, and will continue on until stop codon is found. This is transversion as adenine, a purine, is changed to cytosine, a pyrimidine.